Hello, welcome to our presentation over combating cancer cells with silver nanoparticles. My name is Kyle Leibham.
My name is Sasha Osborne.
My name is Srikar Guntaka.
And my name is Patrick Greenwood.
About the types of nanoparticles used, the only nanoparticle used was silver nanoparticles, but those nanoparticles were all spherical in shape, as shown by this light observance curve. Um, and this is shown because it only has one peak in the curve at approximately 450 nanometers for the light. This shows that the nanoparticles are all around spherical in shape, and the size was approximately 20 nanometers to 50 nanometers. This is shown by the CHEM image on the bottom right of the screen, where some of these nanoparticles are labeled around 20 nanometers, while others are upwards of 50. And there's a little bit of variation, but not much. So it's relatively solid in terms of consistency across the nanometers.
And so, for our application in our article, silver nanoparticles were used to treat cancer cells, more specifically the MCF7 breast cancer cells. And in our article, they extracted the silver nanoparticles from tamarind fruit shells, and they were used to see how those would affect cancer cells. So, the reason that this was prompted and that this was needed to be researched is that traditional cancer treatments, such as chemotherapy and radiation, are very expensive to produce. And on top of that, they are damaging to very healthy cells. So the treatment can be toxic to the patient and overall have many very serious side effects. And so when we look into other treatments such as nanoparticles, they are just as efficient for cell viability because they still decrease the percentage of cancerous cells, but they also increase with the concentration of silver nanoparticles. So they are still as effective, but they are less expensive and not as damaging.
So what exactly did the nanoparticles do? Firstly, the nanoparticles were designed to target and eliminate cancerous cells. The key here is that the nanoparticles left the healthy cells unharmed while selectively focusing the cancer cells and attacking the cancerous tissue. So the specific type of nanoparticles used were silver nanoparticles, and silver nanoparticles were chosen for their specific cytotoxicity, or in other words, their ability to kill cells. This property is essential because it is the cytotoxicity of nanoparticles that ultimately destroys the cancerous cells. So as we look at this image, it shows how actually the cancer cells are being killed. So the silver nanoparticles create something called oxidative stress, which is inside the cancerous cells, and this stress builds up until the cells start to break down and die. So in summary, the nanoparticles target only cancerous cells, weaken them through cytotoxicity, and then through oxidative stress, the cell dies.
So let's talk about the current method of creating and preparing nanoparticles for use. It's a chemical method and it has several advantages and disadvantages. The main advantage being the narrow size distribution. However, it is a high cost operation. It is highly time consuming and energy consuming, and it uses toxic chemicals. And so, for that reason, the disadvantages kind of outweigh the advantages for this method. The other method, which is the one being discussed, is the biological preparation of nanoparticles. And this method is much more eco friendly, it's lower cost. It doesn't require further processing or further steps to complete the processing of the nanoparticles. And it also has a higher yield. This allows the biological preparation of nanoparticles to be a simple one-step process that is eco-efficiency. That doesn't make sense. Is more efficient. It's lower cost, and it's preferable to the current method.
Thank you. Our references was just the one paper by Gomanthi, AC, and et al. The paper was called Anti-Cancer Activity of Silver Nanoparticles, synthesized using aqueous fruit shell extract of Tamarindus indica on MCF7 human breast cancer cell line. We observed that silver nanoparticles hindered the growth of MCF7 breast cancer cells. Thank you.